Hey guys, and for this demonstration, I'm going to be demonstrating the lac operon regulation. So this specific gene has a specific promoter in eukaryotes. So this lac operon has lac construction, so it's for lactose. So as you can see, the P is for the promoter, the O is for the operator, and then the three structure elements to the right are lac Z, lac Y, and lac A. And then to the left is plaque 1 and lac 1. So the idea of lac operon is to break down the lactose, and then the genes to the right over here, lac Z, lac Y, and lac A, produce enzymes in order to break down the lactose. So the Z enzyme helps with the lactose catabolism, which is sugar. Lac Y uptakes lactose from the environment, and lac A also helps with the uptake of the lactose in the cell. So if lactose is present within the cell or the body, then the bacteria wants energy from that lactose. To do this, they have to synthesize the lactose by turning on the operon to produce the digestive enzymes. If the lactose is absent from the body, then there is no need for the digestive enzymes for there to be digestive enzymes for lactose. Then the operon needs to be turned off, making it a positive regulation. These three genes over here to the right are all regulated by one promoter. So this is a polycistronic expression. The upstream promoter is plaque 1 and lac 1, which are the inhibitors. They produce inhibitors that are default or made to be off regularly. So this gene or this segment that is used to digest lactose is regularly off. And it needs to be turned on. So if there's lactose present, it interacts with the inhibitors, and then it changes the structure to stick to the promoter. And then once it sticks to the promoter, then the process of the polymerase can go and transcribe these genes over here in order to be able to digest the lactose. So now that we learned about the lac operon regulation, we're going to be learning about the TRP operon, or the tryptophan operon regulation. So it's a prokaryotic process, and it needs to be and it's needed for proteins and bacteria. So the main promoter, the TRP promoter, and then the O over here is for the operator. So then we have right between the operator and the E, the leader region. We have the leader region, and then the 5 structural genes or elements are E, D, C, B, and A. And these are required for the synthesis of tryptophan. So in order to control this process, we have a R repressor molecule, and this is produced continuously. Once it has been made, it actually can't bind with the operator. So the process for transcribing genes can't continue. By default, the tryptophan operon is always on. This is because tryptophan is a necessary amino acid, so we keep producing it. If there's too much of this in high concentration of tryptophan in the body, we need to turn it off. So what happens is that the tryptophan, once there's a lot in the body, binds to the repressor, and then this repressor becomes active and then can stick onto the operator region right here, and then it blocks the process of the polymerase to be able to transcribe the genes over here to synthesize tryptophan. So it's a self or auto-regulatory pathway in the body. So then, now that we know how tryptophan and the lac operon works, we're going to be looking at the C-AMP and CRP upregulation. So what you need to know is that the preferred type of energy for organisms and bacteria is glucose. And then once this has been taken up, then they will switch to the lactose. This process for metabolizing sugar is always on within organisms. So the promoter area, within the promoter area, there is a cap site right here. And this cap site can also be known as CRP. It's just another name for it. So this site, in the presence of lactose and no glucose, the C-AMP or cyclic AMP will be higher. There will be as there is no glucose, C-AMP is very high. The C-AMP molecule will then bind with the CRP molecule, which is right here, will then bind with the CRP molecule, and this forms a complex which then can attach to the cap binding site, and then once attached, it acts as an inducer to the lac operon. So then it triggers the binding of the RNA polymerase to help the transcription process along, which transcribes the mRNAs to help make the proteins. So with the lactose present, it does not allow the inhibitor to bind, and the absence of glucose starts the just explained process. If there is neither, then none of these processes will happen. Now, when there is both lactose and glucose, as mentioned before, cells or bacteria prefer to use all the glucose first. So they prefer to use all the glucose, and then they will switch over to lactose for energy. But here, because glucose is present, that means that C-AMP here is very low. So they have an inverse relation. If there's a lot of glucose, there's very low C-AMP, and if there's not a lot of glucose, then there's a lot of C-AMP. Because the concentration is very low, then the complex of C-AMP and CRP